sort of the TED-like talks. We're going to have 3 presenters. I'm not sure the order that they were selected to go in, but I'm going to read off the script that I have and introduce each of them. So the first TED-like talk presentation is going to be given by Anya Sondi. The title of her talk is Making the Invisible Visible, Brightening the Target to Reveal and Treat Blood Cancers. She is a 5th-year PhD student in cancer biology.
Cancer immunotherapy, which means I work on ways to help the immune system find and destroy cancer. People picture flashy monitors and breakthrough moments. The reality is mostly me in a lab staring at cancer cells and hoping they behave, which sometimes they do. Sometimes we watch tumors shrink in ways that honestly feel unbelievable, which is what makes the next part so confusing, because cancer doesn't usually come back by overpowering the immune system. Cancer comes back by changing, not dramatically or all at once, just enough so that something which should have been recognized suddenly isn't anymore. Now, imagine trying to find someone in a crowded stadium. They text you, "I'm wearing a bright yellow jacket," so you scan the crowd for yellow, and it works instantly. Recognition feels effortless, but halfway through the game, they take off the jacket. Same person, same seat, same place. But suddenly, you can't find them anymore. They didn't disappear. The bright yellow marker you were relying on did. Our immune system works in a surprisingly similar way. Billions of immune cells are constantly scanning the body in this way, moving through tissues and checking molecular ID badges. Every cell in your body contains tiny molecular identifiers that signal whether it belongs. Your body survives by distinguishing self versus not self, healthy from dangerous, and belonging from threat. But occasionally, something slips through. Most of the time, this system works beautifully. It clears infections, it removes damaged cells, it even eliminates many early cancers before we knew they were there. But here's the problem. Every time a cell in your body divides, it has to copy its DNA. All 3 billion letters of it. This process is unbelievably accurate, but not perfect. Sometimes a tiny typo appears in the instructions. A mutation. One mutation usually doesn't matter. Cells have safety systems that fix mistakes, backup systems that shut damaged cells down. But every once in a while, one slips through, and a cell may accumulate just enough changes that it starts ignoring the rules. It can stop, when it is told to stop dividing, it keeps going. When it is told to die, it survives. And quietly, slowly, that cell begins to grow into cancer. Now, scientists have wondered for decades, if we can teach the immune system to recognize disease, could we teach it to recognize cancer better? This idea led to one of the most remarkable developments in modern medicine. CAR T therapy. CAR T cell therapy is where we take a patient's own immune cells from their blood, specifically T cells, the immune system's most powerful fighters. In the lab, scientists genetically engineer these cells. We add a special receptor called a chimeric antigen receptor, or CAR. This receptor is specially tuned to teach the T cell to recognize a target on the cancer cell. Once we've genetically engineered these cells, we grow millions of them in the lab, and then we return them to the patient's body. But these cells are different now. They aren't just circulating, they're searching. They're scanning every cell they encounter for that molecular signal. They're powerful hunters, and they can eliminate tumors the size of a golf ball within weeks. CAR T is one of the first treatments in modern medicine that is alive. It is a living drug. It is so advanced that even some cancers at advanced stages may disappear completely. Now, imagine someone lit a signal, a fire burning in a remote area at night. Rescue teams are trained, they're equipped, they're powerful, but they still depend on one see the flames. If the flames are bright, the response is fast and precise. But dim the flames even slightly, and the source becomes harder to find. Not because the rescue team is weak, but because the signal is faint. In mantle cell lymphoma, the blood cancer that I study, cancer cells don't usually erase their molecular markers completely. They lower them. They dim the flame. CAR T cells in this disease are taught to recognize a special receptor called CD19. CD19 is a protein found on the surface of many blood cancer cells. You can think of CD19 as a flare on the surface of the cancer cell. And if the flare is bright, the CAR T cell has no trouble at all finding the target cell. But dim the flame even slightly, and recognition is difficult. CAR T cells don't fail because they're weak. They fail because the cancer stops showing its face. Mantle cell lymphoma cells reduce the amount of CD19 present on their surface in an effort to hide. Now, we saw a question in our data saying, what about this fire makes it burn so bright? What about this CD19 expresses the amount that we want on the surface? So this led us into the internal machinery of the cell, into the transcription factors that determine which genes are active. And one of those transcription factors, called IKZF1, kept appearing in our data. Now, IKZF1 may sound like a password that you'd forget almost instantly, but it plays a key role in cell development and differentiation. So we tested it. We increased IKZF1 levels of protein in mantle cell lymphoma human models, and then we asked two questions. One, do the cancer cells become easier to see? And two, do the CAR T cells respond any differently? Now what we saw was striking. When we increased IKZF1 levels, CD19 increased. And when CD19 increased, the CAR T cells performed better. It was like watching a miracle happen in real time. But still, this development was not translational. We needed a way for this to be able to help patients versus only raising IKZF1 in a lab. So this led us to a drug called slomitasertib. Slomitasertib is a drug that blocks CK2 alpha, which is a protein that normally keeps IKZF1 suppressed. When we use slomitasertib to block CK2 alpha, IKZF1 stabilized, and CD19 came back. It was like watching the immune system stop swinging in the dark and finally land a clean hit. And I think that's the part that keeps me coming back to the lab every day, even when it feels like the cancer is always one step ahead. Because every once in a while, you get a graph that tells us something is working. We're catching up. So instead of sharpening the sword, why don't we try turning the lights back on? Sometimes we don't need a stronger weapon. We just need a clearer target, because for decades, scientists in immunology have focused on making stronger CAR T cells, making them more aggressive, making them last longer. But what if all we needed was to turn the lights back on? In the beginning, I asked you to imagine someone taking off their yellow jacket in a crowded stadium. Cancer does something similar, but it doesn't always disappear. Sometimes it just blends into the crowd enough by turning down its signal. Our work is focused on turning that signal back up, because the immune system is already incredibly powerful. Sometimes it doesn't need a new weapon. It just needs the lights turned back on. Thank you.
Our second presenter for our TED Light talk is Philip Servan-Dragan.
Yes.
The title of his talk is Application of Generative Artificial Intelligence in Teaching Preventive Science. He is a second-year PhD student in prevention science and community health.
Hello everybody. Today I'm going to answer the question why most education never gets used and how artificial intelligence can change that. Let me start with a question for you all. How much of what you have learned in school are you actually using? I'm not talking about how much you remember, I'm talking about what you actually use in your day-to-day activities. Come on, make some guesses. It's really interactive.
2%.
2%? Okay. Who offers more? You, come on.
A lot more.
A lot more.
Yeah.
Okay, but we don't know how much. I want to hear you. Okay. 5%. Okay. The truth is that 37%, which is surprisingly low compared to how much knowledge we gain every single day. And a study conducted by HR Block with more than 2,000 participants also found out that 84% of these participants said that they have learned things in school that they never ever got to use in their day-to-day activities. This should stop us right there on track and make us ask, what are we doing? What are we teaching? If education is not being transferred into the real world, then there is a challenge there. Something is not going wrong, not going right. So we know that something is missing. The truth is that we are so good at teaching information, but far less good at teaching application. And that shows it. When we look at experiential learners, these are people who learn differently, learn by doing, hands-on. They are 71% of experiential learners say that their degree is relevant to their work, versus 56% of non-experiential learners say that their degree is relevant to their work. Wow. That gap is the difference between knowing and doing. And nowadays, this is more consequential than in public health and in prevention science. Because prevention, we aim to prevent bad stuff from happening. Prevention is about communities, hospitals, people who are dealing with challenges. You basically don't prevent addictions and mental health problems by memorizing definitions. No, you do it by being in the real world, by being hands-on, by talking with people. So we ask ourselves, what can we do? What can we do to solve this big challenge that we have? We have a challenge. There is so much knowledge that is not being applied. We ask ourselves this research question. In my research lab, we ask, what if our classrooms look more like the real world? What if instead of just loading their brains with information, we make it more interactive, more engaging? Before going into the solutions, let's look at how currently the educational system looks. We have the traditional method. I bet we are familiar with this one. You are presented with a theory, then some concepts, some examples, if time allows, and then some tests. ABC, multiple choice. Prevention is not like this. No one is going to call you and tell you, hey, I have a problem. What's that theory that you have learned in school? How can I apply it? No, a mayor is going to give you a call and is going to tell you about raising alcohol rates in his community. A school principal is going to write you an email. I need help. Bullying problems are knocking at my door. And that's more closely related to the flipped classroom method. In the flipped classroom method, students are presented with a situation. Only there, after they understand the situation, they reach out for theory. Together they come up and create solutions that they present to the community. Well, that's what we want to do. We want to create solutions that use this flipped classroom method. And we came up with two of them. Number one, we use artificial intelligence to create case studies. Case studies that are inspired by reality, where students can see concepts that they have learned in the classroom in the story form. And second, we use artificial intelligence to simulate interaction. First, we say to create case studies using the Zapier platform. It's a platform that allows you to create a chatbot. We loaded that platform with our course content, prevention science and community health. And we created something called Prevention X. This is our chatbot, our little baby. And we went to this little baby of us and we gave it a prompt using only what we loaded you with. So stay in these boundaries. I want you to generate me a case study on distraction. Person, age, nationality, socioeconomic status, risk factors, protective factors, health issues. Are you curious to see what it generated? Let's look at it. It generated 4 of these different personas, different stories. This is just one of them. Amina is a 20-year-old Somali woman living in a refugee community in mid-sized city in the United States. Her situation is compounded by limited access to education and job training programs. On the positive side, has strong familial bonds. This is just a small example. Highlighted with yellow, you can see concepts that we have taught in our class. Risk factors, protective factors, social determinants of health. This time, students were not just memorizing it. Were presented with this case study and together in teams were asked to respond to the following questions. What adverse health outcomes do you see? What protective factors do you see? What social determinants of health are currently at play for me? How did they react? Did they like it? Did they not? We asked them, how do you rate your experience answering this question in a team format? They did it as a teamwork. Most of them said good and excellent, with some of them saying okay, bad, and terrible. This is the first step. The question is, can we take it one step further? Can we make it even more interactive? In an ideal setting, I'm going to take you, all of you, with my classroom of prevention science student, go to the mayor here in Miami and tell her, hey, we have these theories that we have found out. Talk to us. Tell us your problems and we're going to come up with solutions. In an ideal setting, I can do that. But the money doesn't allow it currently. It's not feasible. So what if we make our classrooms to make more like the real world? What if instead of you all going to the community, going to the school principal, going to the mayor here in Miami and using what you have learned in the classroom, what if we bring the community here? Is that possible? Let's see. I want to introduce you to two of my friends. They became so close to me. He is Mayor Francisco. Mayor Francisco is a mayor of a small city here in Miami that is dealing with raising alcohol rates. He tried everything to solve it. People are drinking and driving. There are so many problems in his community and he needs to find a solution. And then we have Marisol. She's a school principal at the school here in Miami, rising bullying rates in her community, in her school, emotional problems. She tried everything to solve it, but she couldn't solve it. What we did, we created these AI-generated personas. We gave them the roles, we gave them the identities, the tone. How should you speak? What should you reply? What do you know, what you don't know? What questions are you asked to ask the questions? And then we presented to the students, and this time it's important. Focus up. They were not just students. We gave them a role too. Students, you are a prevention science expert. You are an advisor. You are a counselor. Use what you have learned to provide feedback. Students were not memorizing definitions or multiple choice ABC. No, they're asking questions, proposing solutions, receiving feedback. This is real time, like the real world. Curious to see if it worked? Well, let's see. We asked our students, compared to the additional prevention lectures, how did the interaction with the AI-based persona make you feel? Much more engaged, somewhat more engaged, with about the same and less engaged. The majority of them said that it was more engaging. Then we asked them, how realistic did the interaction was? And they said, very realistic, somewhat realistic, a little of them saying just a little realistic. Then we asked them, how much did the AI interaction help you understand how to apply prevention science concepts in the real world setting? Remember that application? Just 37% is applied. Let's see what it said. Very much, somewhat much, with just a few of them saying a little. And lastly, we wanted to see how confident they felt after interacting with this AI-generated personas. After interacting with the persona, how confident you were, much more, somewhat more, and the confidence level stayed the same for 28% of them. We have these things. Are you curious to see one of these examples of the interactions between the students and the AI persona? Okay, let's look it up. So, hello. I am Marisol Pierre, principal of Rural Middle School here in Italy. Thank you for taking the time to really think about what our students are experiencing. Ask me anything you need to better understand our students. And our student is asking the following question. What challenges do your students and community face? She responds, our families are dealing with housing instability. Overcrowded living conditions, which can make it hard for kids to focus or even get enough. On top of that, language barriers, teachers burnout. There's a lot. She seems overwhelmed. But she has the power to ask the following question, to engage in the conversation. Does that help you get a picture? What part of this feels most important to you? The students reply, that sounds tough. It makes me wonder why staff keeps coming back. Do you guys feel that you have helped kids navigate a lot of these situations well? The students collect information in order to propose solution later. She said this thing, you do see positive moments, the students grow, but it's a mixed bag. I'm curious, when you think about helping students, what kind of support do you imagine, do you envision? The school principal is very proactive, searching for answers, and the student came up with a solution. I think some sort of campaign to let the students know that the support is available. Like even for them to just have the knowledge that if they ever struggle with something personal in the future, there is help there. Also, perhaps an anonymous page where they can submit concerns, and also webinars where they can be taught how to deal with such an issue. This is a newly invented solution by our students who participate in the classroom. It sounds innovative, it's creative, and it's actually very productive. It can work. And the school principal provides feedback. She says that your campaign to spread the word, support is available, sounds like it could be helpful. She likes it. Then the anonymous form is interesting too. Sometimes kids don't want to be signed out or worry about how they will be seen by peers. So having that anonymity helps. This is just a fragment. The conversation continues for 15 more minutes where they get more into depth on who is going to implement these webinars, when it's going to be, how are we going to convince the parents to come. It's hands-on, it's interactive. It's awesome in my opinion. Let's see the impact. The impact is clear. It's much more engaging than just sitting in a lecture. It's also realistic, and it helps their understanding. You could see that they feel more confident in their knowledge and their understanding is improved. Now, we have some limitations. The sample was small. Among our studies, we have from 21 and to 76 participants. We had also mixed opinions. Some students, when we asked them in an interview in qualitative text questions, how did it feel for you to interact with the AI? Some of them said, I felt engaged, helped with brainstorming, while some others said, it's overwhelming, too many questions. I feel that the AI is too pessimistic. And it can feel like that, but sometimes the school principals are like that, because they are tired. You know, they tried everything. And lastly, it's the technology part. Some students say that they would like to see more visuals. Maybe in the future, UM is going to give us funds to create a cyborg, you know, with the face of the mayor here in Miami. Who knows? What I'm talking is mostly how do you prevent students that interact with AI to use AI to respond to the AI. We had some students who used ChatGPT, copy-pasted things from the chatbot in the ChatGPT and then copy-pasted from ChatGPT into the chatbot. So we don't have a solution for that other than training the chatbot to send a message. Hey, I'm interested in your opinion. So please, use your brain. Smiley face when you interact. That worked for some of them. So these are some quotes when we ask them, how did it feel? I felt like a preventionist during the AI interaction. Some of them said, I related to the things I said. I learned it as a science teacher to middle school students. This person was actually in the field, so we received feedback from someone professional who said that it's realistic. And some of them said, I did enjoy this activity and I think it would be helpful, but still I think it is a little bit more work to be able to flow the process much more seamlessly. The truth is that currently AI is offering something that education always struggled with, and that is a safe practice. It's a safe environment where students can apply what they learn without hurting anyone. AI is finally offering realistic complexity and immediate feedback for the solutions and ideas that students have. I'm not proposing a replacement of you all professors and teaching assistants. No, relax. You still will keep your job. What I'm saying is that if we want students to use more than 37%, if we want them to feel confident and to say that their degrees matter, and they are ready to go in the real world and make an impact, feel confident interacting with change policymakers, stakeholders, then we need to bring AI into the classroom. Not to replace the classroom, but to extend the classroom, to bring the classroom into the real world, into the interaction, complexities, into this continuous feedback. And by doing this, by bringing the AI inside the classroom and extending the classroom in the community, we can actually have the most powerful prevention strategy at all. Thank you so much. Academy of Medical Education Scholars, UM Platform for Excellence in Teaching and Learning, for inspiring us, and the Department of Public Health Sciences, my advisor, right here, is Dr. Eddie Brown, And of course, I want to thank God for inspiring me and giving me another day to be alive to present my research. Thank you again. That's my time. Reach out to me.
All right, our third presenter, our third and final TED Talk like presenter is Indranee Mukherjee. The title of her talk is When the Placenta Turns Hostile, Decoding Oxidative Stress in Preeclampsia. She is a postdoctoral scholar in infectious disease. Do you want to Can you hear her in the back?
Okay. So before I start, I want to ask you a very quick question. How many of you here have been pregnant or have heard of people being pregnant? Can I see show of hands, please? Yeah? And keep your hands up if you guys have heard of preeclampsia or toxemia. See? Exactly. So 1 in 12 people in the US, they suffer from this mysterious condition, and yet many of us here have never heard of it. Hi, I'm Dr. Indranee. I'm a postdoc in the Department of Pediatrics in Miller School of Medicine, and today I'm going to tell you a detective story. So the story unfolds around an organ which is the placenta, a very, very amazing, fantastic organ that really grows a human. And let me paint a picture about what is preeclampsia. Imagine you're pregnant, you're 6 months pregnant, everything goes fine, everything is happy, your baby is happy, doctor said all good, but suddenly, bam, your blood pressure shoots up, and suddenly you start seeing spots in your vision, and you spill protein in your urine. What's happening? Like, you're confused, right? This is preeclampsia. And preeclampsia can occur due to multiple causes. There is no one such cause that is responsible for this disorder. It can lead to seizures, it can lead to stroke, and in the worst case, it can even cause death of the mother and the fetus. So, and you know what? The only cure that we have till date for preeclampsia is delivery. No, the baby is not the problem. The problem is the placenta. And preeclampsia has majorly two facets. Think of it as identical twins who look similar, but they are very different in how they behave, their identity. So there goes early onset preeclampsia and late onset preeclampsia, the two twins. So what happens is, in early onset, this is said before 34 weeks of pregnancy, and it is, you can say, it's the troublemaker. You know, it does not let the placenta form, let alone function properly. And the second twin, the late onset preeclampsia, it is the sneaky one. It will let the placenta form, but then it will play with the mom's blood vessels, or it will do something to cause harm to the mother and the baby. So my research is majorly focused on early onset preeclampsia and to understand what is going on with it. So now, don't be scared, I'm not going to quiz you, no using technical jargons, this is just a picture which will make things easier. So imagine that you are trying to water your garden and you are using a garden hose. How easy will it be, right? So look, in normal pregnancy, we have something called spiral artery, which is there in all the females, all of us. But the spiral artery thickens and broadens when a lady becomes pregnant. So what happens is, blood flow through the spiral artery is easy, just like watering your garden through a hose is easy. But in cases like preeclampsia, this hose, instead of the hose, you can just imagine watering your garden using a straw. So what is going to happen is, the blood flow through it will be very shallow, irregular, turbulent, and choppy. And this scenario, when it happens in a pregnant woman, it gives rise to a condition known as ischemic reperfusion. Now again, science is very fancy, we use terms, but let me explain. So suppose you have a fireplace and you have fire. So what will happen is you will have tiny sparks coming out of the fireplace. So when these sparks come out in controlled manner, they are an indication, right, that hey, there is fire. The fireplace, there's fire and it is good. You know that the fireplace, the fire's on, right? But when these sparks are very regular and they're coming out so often that they're burning the curtain, sofa, carpet, that's not done. That's uncontrolled condition. And this exactly is what is reactive oxygen species. So just imagine the sparks, they are reactive oxygen species, the chaotic molecules, and the environment created is known as oxidative stress. So here comes my detective question, that what is reactive oxygen species doing to cause this disorder, preeclampsia, and also to find a mechanism. You know why we need to find mechanisms? Because if I know what is the pathway that ROS is triggering, we can find a drug to interrupt the pathway and to stop the disease progression. So, again, not to scare you, I know there's a lot of arrow and there's a lot of boxes, but this is just to show that we did our homework correct. And also in science, you have to use multiple ways to come to a conclusion. So, what we did in the lab, we took a bunch of placental cells, we tried to generate those sparks or oxidative stress in a controlled way. And then we compared it with the patient samples, our PE samples. Interestingly, this is the first set of results that we found. So see, this is how the placental cells look in the lab. And as I mentioned to you, we were trying to treat them with different amounts of the stresses to cause the chaotic environment, the spark environment. And the arrows that you see here are the cells which are saying that, hey, stop, this is too much of stress, we are dying. So it had to be absolutely, it had to be like Goldilocks, not too much, not too less. So we kind of ended up choosing an amount where the cells can take it, but the amount will not end up killing all the cells. What's the meaning? If the cells die, we will not even be able to understand what the sparks are doing, let alone find any cure for preeclampsia. And again, don't be scared, too many colorful graphs, I know. Just to remind you, I won't quiz you. This is a proof that yes, the dose that we chose or the amount that we chose could generate ROS or create an environment which can mimic preeclampsia-like environment in real pregnant women. Moving forward, placenta, it is very interesting. It's made up of different cell types and every cell type has a different function. Imagine you're in an office and the office has different departments, right? Each department having an individual cell of work. That's exactly how the placenta functions. And there is a function known as differentiation. What does it mean? It's very, very simple and easy. So placenta is the organ which connects the mother and the fetus. So differentiation is the process by which we can build a wall, brick by brick. Fuse the cells together so that there is no mixture of mom's blood and baby's blood. And when we created ROS-like environment, we found that differentiation was strikingly going down in the placenta cells, indicating that definitely ROS was one of the key players contributing to this dangerous disease. But, and the key player in this differentiation is syncytin 1. Now, I'm going to tell you a very, very fun science fact. Syncytin 1 is a viral gene. So, millions of years ago, what happened? One of our ancestors had some kind of viral infection, and the virus, it inserted its DNA into our genome. And we kind of decided to keep it. Why? Because we are going to use it when we will make babies. Evolution, y'all, And so, long story short, this protein is the key indicator that the cells are differentiating or forming the wall which is dividing the mother's and the baby's blood, and ROS was definitely making its levels go down. And when I mentioned that it has different departments, the placenta has different functions, invasion is one of them. What does it mean? I mentioned to you that the spiral artery has to be, remember plumbing, it it has to be very broad for the blood to flow and not like straw through which the blood flow will be choppy and turbulent. So that is done when the cells innovate into the maternal wall. But when we wanted to see what's going on with this particular department, we saw that innovation was strikingly going up when there was this spark-like situation created in placenta. So, is that not good news? Because I just mentioned that that's what we need. But to our utter astonishment, that was not some good result. You know why? Because this was happening in an uncontrolled, chaotic manner. And we want every function to happen with peace. We don't want the departments to fight, at the end of the day when we get salary, do we? No. So this is how the cells look. I know that many of you are not familiar with seeing cells under the microscope. And this is how the cells look. They look very, very beautiful. And next, we wanted to see what's going on with the cells internally. It's like you're zooming into somebody's gut to see how is it, what is it made up of? How does it look like? And this is done using something called transmission electron microscope. And when we wanted to see, so you see the TP stands for term placenta or healthy placenta, and PE stands for preeclampsia. When we wanted to see what's going on, we found that the lipid droplets were so high. All of us have heard of lipids, right? This is how it looks under a microscope. I mentioned to you that blood vessels are so thin in preeclampsia that the blood flow from mom to the baby is hampered. Look, the blood vessel is so thin. And also, we all have heard of glycogen or sugar, right? Sugar content. Look, the cells were trying to save sugar as much as possible in preeclampsia compared to term. The arrows, they indicate that. What does it mean? It means that there is some serious issue going on and the cells are trying to prepare for a war-like situation. The environment has become so hostile. And in continuation, when we wanted to look into some other structures like mitochondria, which is usually known as the powerhouse of the cells because it produces the energy that I need to speak here, the energy that requires you to sit here and look at my presentation, that is provided by mitochondria. And look, in preeclampsia, we found that this mitochondria is so black, it's literally gone, you can barely see it, its function is lost. And same is the function with endoplasmic reticulum. It is basically the protein folding factory of a cell. And here we ask the question, is there any mechanism that we can trigger? This needs to stop. The entire environment has become so hostile. This is a war-like situation. And there, the answer was UPR or unfolded protein response pathway. Again, a fancy science term, nothing to be scared of. Imagine you have washed clothes and you have taken them out from the dryer and they are all misfolded. They are all here and there. So what will you do? You will stop all your work, first fold the clothes and put them in the closet. And that is exactly what this pathway does. But in preeclampsia, when there is this alarm that, hey, please fold your clothes, what's going on? Nobody was there to fold the clothes. And that is what the pathway was. That this pathway, which was responsible for folding the proteins or making the environment back to normal, was lost. And that was done through one of the arms of the pathway. So, in short, to conclude, we could find out the main cause for this dangerous, mystical disorder, preeclampsia, which is ROS, which was majorly causing changes to its functions of the different departments of the company here, placenta. And we could also find out that the pathway, ROS induced ER stress, induced UPR pathway, and which was very helpful because now we can think about treatment, we can think about interrupting the pathway and stopping the disorder from spreading further. Since I think the viral protein that I told you was going down, and all of this was causing high blood pressure, which I mentioned to you in the beginning of my talk, that is one of the major symptoms of preeclampsia, and this is why we see it in the mother. So now what? We found the pathway. So what's going on? So the interesting fact is we can now make changes. How? By reducing oxidative stress, which is this pathway condition that we generated and is usually seen in the preeclampsia placenta, by reducing the ER stress. Remember the protein folding pathway trigger? We can reduce that stress. How? By making lifestyle changes. We can eat good. We can try not to have sugar. We can have reduced triglyceride levels. And also, we can try to use some of the drugs that's already there to mitigate the effects of that protein folding or pathway or UPR pathway in preeclampsia. You know what will happen? This way we will save years of science, years of research, because the drugs are already there. All we need to do is use it in dissolve. So, science can never be done alone, so I'm very thankful to my mentors, Dr. Rana Chakraborty here at U Miami, Dr. Shubhradeep Karmakar at AIMS, All India Institute of Medical Sciences, India. All my lab mates, all my kids sitting here, they have all been very helpful. And let's be honest and real, science cannot happen without funding, so I would like to thank my funding agencies, NIH and CSIR. And I would like to thank each and every one of you for listening to me, and you can ask me any questions. Thank you so much.